本节目由汉康生技企划制作，以平台转移生命希望，布局全球商业价值。汉康生技驱动抗癌革命的领先基因。各位听众朋友大家好，欢迎回到与汉康读懂肿瘤免疫新药的抗癌革命。在前面的集数，我们从创办人的愿景、商务开发韧性，聊到了工业生产的硬实力。但是对于生技投资来说，最终决定成败的战场始终是在临床试验的数据表现。那么今天的来宾呢，也非常的关键哦。我们邀请到的是汉康生技的集团总裁及医疗长陆英明 Alvin。他的任务呢，是把实验室里的科学转化成为人体临床实验上的治疗价值。最近汉康首度对外公布了 HCB101 在胃癌二线治疗的三药联合策略，以及在头颈癌单药治疗上看到的惊人数据。今天就想跟听众们分享，究竟这些数据对于患者意味着什么？对投资人的资产增值又代表什么样的意义？ So today's guest is crucial to this discussion. We are joined by Dr. Alvin, President, Chief Medical Officer, CMO of Hong Kong Bio and CEO of US Hong Kong Biopharma. Alvin's vision is to translate laboratory science into tangible therapeutic value in human clinical trials. Recently, Hong Kong Bio disclosed for the first time its triple combination strategy for second-line gastric cancer treatment using HCB101, as well as astonished data observed in head and neck cancer monotherapy. Today, we want to share with our listeners what exactly do these data points mean for patients, and what do they represent in terms of asset appreciation for our investors. Welcome, Alvin. Alvin, say hi to our audience.
Hello, everyone. 你们好，我是 Alvin. Well, thank you for having me. I will speak in English today because my Chinese may sound a little unusual, 很怪的, but I'm very happy to be here with all of you.
Before we dive into the dialogue, I would like to quickly decode 3 key terms to help our audience understand the high stakes involved today. The first of all is SOC, Standard of Care. This is the most effective treatment currently recognized by the medical community. If a new drug can challenge the SOC, it has the potential to become the new market benchmark. The second one is RO, Receptor Occupancy. Imagine the surface of cancer cells have many parking spaces, and the drug is the car. And RO more than 95% is like a full parking lot. It means the drug has completely locked onto the target, reaching its peak biological activity. The third is PR, Partial Response. Simply put, this means the tumor diameter has shrunk by more than 30%. In medical imaging, this is an extremely clear and definitive signal of efficacy. Fighting the solid tumor with just one drug is often very difficult because this tumor builds strong defenses that block medicines from getting inside. That's why Hong Kong Bio uses a golden triangle combination strategy approach. So, Alvin, Hong Kong Bio recently disclosed a powerful triple combination strategy for second-line gastric cancer, integrating HCB101 and a VEGFR2 antibody, and the chemotherapy drug paclitaxel. From the clinical design perspective, why do we choose this specific triple treat combination instead of just a doublet? How does the triple immune attack, which simultaneously unmasks the tumor, cuts off its supply lines, and delivers direct cytotoxicity, create a synergistic effect that is greater than the sum of its parts to challenge the current standard of care, SOC?
Okay, this is a very important question. Why a triplet? When you're talking about the second-line gastric cancer, is still a very difficult setting. But by the time the patient gets there, the cancer has already come back after first-line treatment, and it usually becomes more aggressive. It also finds different ways to escape the treatment. So our thinking was not simply that more drugs are better. This is not the point. The real question was, can we address several problems at the same time in a way that is scientifically logical and still tolerable and safe for patients? If you break down this regimen like what you just said, Vivi, earlier, each one has a different job. Paclitaxel is a chemotherapy, and it is a direct cytotoxic agent. It directly kills tumor cells. And doctors already have a lot of experience using this in this setting. The VEGFR antibody you mentioned helps change the tumor environment by affecting the tumor vasculature and by shifting some of those immunosuppressive signals that comes with the VEGF biology. So in simple terms, it may help make those tumors less protected and less supportive of cancer growth. And HCP101 is designed to help the innate immunity, especially macrophage. Remember I told you that this is the upstream. They do the job better, recognizing, attacking, and helping the immune system respond to the tumors. So when we talk about synergy, we don't mean it as like marketing or like a slogan. We meant it in a very practical clinical sense. Do we see response that are deeper, happen earlier, and look more consistent without creating an acceptable safety problem? That's really the hypothesis that we are testing. A doublet, two combination, may address one or two of those problems. A triplet is our attempt to block several escape routes at the same time. And of course, the early observations are encouraging so far, but they are still early. In the end, what really matters is whether the benefit lasts and whether we can see the same pattern in more patients.
Yeah. Now I'm curious because before HCP101, there are so many medicines that used to treat the cancer patient or their tumors. So what is the potential of HCP101? Because there are so many medicines, we can combine any other three medicines to treat cancer. But I think HCP101 have its potential. What is that?
Yes, and honestly, this question, I think that exactly the right question, Vivi. There are already many cancer medicines. And in theory, you can combine many different drugs. So the real issue is not whether we can combine 3 drugs or more drugs. The real issue is whether one of those drugs brings something truly different and truly useful. So what makes HCP101 interesting is that it works through a different part of immune system. Remember, the innate immunity. Most current immunotherapy mainly focus on T cell, right? Like PD-1, antibody. All of those are adaptive immunity downstream. So you have to have the cell macrophage to attack. If you break this upstream system, it's already not working. Especially if you're talking about PD-1 pathway, which downstream. So HCP101 is designed to engage the innate immune system, particularly macrophage. Again, macrophage is the cell to attack tumor, to help recognize and clear those tumor cells. So the potential of HCP101 is not just it can be added into combination. The potential is that it can activate another layer of the anti-tumor immunity. And if that continues to be supported by our clinical data, HCP101 could become a strong partner for other therapies like what you just mentioned in the introduction, like chemotherapy, target therapy like HER2 positive like Herceptin, or existing immunotherapy like PD-1, PD-L1, because it brings a complementary mechanism rather than more of the same.
Yeah, and I saw the data. The data shows that within just 6 weeks of treatment, gastric cancer patients receiving this triple combo saw a comprehensive shrinkage of systematic metastatic lesions. The word lesions, which for our audience, simply means the areas where the cancer has spread or formed tumor. So how rare is this simultaneous response of both target and non-target lesions in the current clinical landscape? And how does this position Hankoh Bio to lead in this $4 billion market?
Oh, okay, the 6 week. So this 6 week imaging scan, what is really interesting in the overall is the pattern. Don't forget about this pattern. We are not just seeing our tumor spot, what you call lesion, getting the cell smaller in diameter. That's correct. But we are seeing multiple lesions shrinking at the same time, not just one spot. They're telling me that and suggesting the combination may be having a broader or more systemic effect, should say that way. At the same time, I will be careful not to overstay early imaging. A scan at 6 weeks, again, 6 weeks can be very encouraging. But the real questions are, does this response go deeper? Does it last? Or do we have the same pattern in more patients? That what turns an early signal into something clinically meaningful. So yes, Vivi, if this kind of effect continues to hold up with durability, consistency, and manageable safety, then it could potentially raise the bar in second-line gastric cancer. As of today, in our middle dose, you can see 5.12 and 8 milligram per kilogram, all 10 second-line gastric patients respond. This means something. The pattern is consistent. So as on the business side, I understand the marketing, the commercial opportunity is of course important. But I always come back to the patient's need first. This is still a very difficult disease. And second-line outcomes remain limited, or the outcomes are modest. If we can show better and more durable benefit in that setting, then the broader market's impact will follow naturally.
Okay. Beyond the gastric cancer, we've seen very encouraging signal in first-line treatment for head and neck cancer. And with HCP101 monotherapy alone, tumor diameter decreased by 42% PR for more than a year. So, Alvin, this suggests a major pivot. HCP101 is moving beyond just indication-specific efficacy in head and neck cancer to a broader strategic positioning across various tumor types and treatment settings. Since current therapies still rely heavily on PD-1 inhibitor, how do you see HCP1's mechanism positioning it as a future backbone therapy, perhaps even in earlier treatment?
We move to the head and neck. Yeah, that head and neck signal definitely caught our attention. We saw a durable partial response lasting more than a year, 53 weeks, with HCP101 monotherapy, which is something we take very seriously. Now, just a reminder, Vivi, none of the early CD47 targeting agents show anti-tumor activity with monotherapy in solid tumors. So just to clarify one point about the line of therapy, in Taiwan, many patients still receive chemotherapy as their first-line treatment because PD-1 drugs are not always accessible. So in Taiwan, these patients are technically second line. But from the disease biology perspective, they are quite similar to first line recurrent or metastatic head and neck cancer patients in many other countries. So even with that context, the signal is still encouraging. But we also have to stay careful. Or these are still early data. So the right way to think about it is as an important signal that needs to be confirmed in more patients. So what makes HCP101 interesting is the biology. Most current immunotherapy, as I said, especially PD-1 inhibitors, mainly focus on where? The T cells, the downstream, the adaptive immunity. HCP101 is designed to engage the innate immunity system.
Yeah.
So the upstream, particularly macrophage, those to attack the tumor cell, which help to recognize and clear tumor cells. Because of that, there is a potential, really a potential, that HCP101 could have a broader prevalence across different tumor types. But of course, broad does not mean everybody. Just like PD-1, it's not universal for every patient, you know. So the key question is, where this biology matters the most, and which patients are most likely to benefit.
Yeah.
So in terms of the combination, this may actually be where the mechanism becomes very powerful, Vivi. If HCP101 is an innate immune moderator, it can logically pair very well with, again, chemotherapy, HER2 positive antibody like Herceptin, or VEGF antibody like ravulizumab, or even PD-1 treatment. And then it could potentially become part of the broader treatment backbone. Now, when I talk about a potential backbone, I mean it combines cleanly, it is tolerable and safe, and it adds benefit across different regions. And for the early treatment setting we're talking about, that is something we think carefully about it. Remember, as an early disease may offer a better biological opportunity, like bigger market too, because the immune system is often more intact. But there's a huge but. The bar and most of those historical data and the standard therapy is also higher because the current standard therapy already work well there. So we should only move earlier if the overall clinical evidence, again, safety, consistency, and durability, how long it lasts, clearly support the risk and benefit balance.
And I'm aware that there are already many anti-cancer drugs on the market, some targeting innate immunity and others targeting adaptive immunity. Since HCP101 acts as a bridge between the two, if future scientists combine two separate drugs, one for innate and one for adaptive immunity, along with a PD-1 inhibitor, would HCP101 still maintain its competitive advantage?
Okay, so now you're talking about, can you combine all the different drugs? No, Vivi, this is a very thoughtful question.
Yeah.
In theory, yes.
Oh.
Scientists can always try different combination of separate drugs.
Yeah.
This is how oncology keeps moving forward. But in practice, more drugs do not automatically mean a better treatment, or more mechanism on paper do not always translate into a better medicine in real life. We know that. So the real question again is whether the biology works, whether the combination is tolerable, and whether it is practical for patients.
Yeah.
They are very sick, and for doctors to use. So what makes HCP101 interesting is that it's designed again to connect the important part of the immune response in a single approach. So the potential advantage is not only the mechanism itself, but it also the simplicity and the ability to combine with other established therapies in a clear and cleaner way. If in the future someone uses separate drugs to bridge the innate and adaptive immunity like us, that could certainly be explored. But for now, HCB101 is the only one. But combining more drugs also often means more complexity. In where? In the safety. Dosing more complex, development will be more complex, and will be more cost because you added more drugs. So I think the competitive advantage of 6CB101, it continues to be supported by the clinical data, is that it may offer a more integrated and practical way to activate the immune system, rather than building increasingly complicated combinations step by step. In the end, the real advantage always comes back to the clinic and patient. Does it help patient more? Is it safe enough? And is it easy enough to use in real practice? That is what will decide.
So, how is Hanko Bio structuring its global clinical development strategy to ensure alignment with the highest regulatory standards across major markets?
So the basic idea is actually very simple. Our approach is to build the program in a single global standard from the start, not a different strategy for every region. In practice, that means designing trials, the endpoint, the safety monitoring, the data system to meet the expectation of the most stringent, demanding regulators, such as what? The US FDA, the EMA, or broader ICH standards. In fact, I have already upgraded the system to global standard. Because in the end, regulators are not only looking at the drug itself, they are also looking at how strong and how reliable the evidence is. We also spend a lot of time on execution on the operation side. That means things like consistent imaging review across Taiwan, mainland China, and United States, clean source document from the site, and clear rules for dose escalation, expansion, and go-no-go decision. Those details may sound operational, but they really matter because agencies like FDA, EMA, or even the Taiwan FDA will do inspection. This is why I personally visit sites in Shanghai and Guangzhou to do self-inspection in general. And yes, there can be ways to move faster across regions, but only if you do it carefully. The goal is not to run separate studies and create more complexity later. The goal is to generate one strong data package that can stand up globally. I have learned that if you try to save time by cutting corners earlier, you usually lose time later. So for us, the fastest path is often the most disciplined.
I understand that Hanko Bio have shown very promising results across various clinical trials, and I was wondering about the patient demographics. Were these participants exclusively Asian, or did the trials also include American or individuals from other countries?
Wow, the Asian. Well, this is a very important question, Vivi. So far, most of the patients in our study, of course, have been Asian. But the program itself is definitely not limited to Asia. We are running the international clinical trials across Taiwan, mainland China, and the United States. None of that reflects the reality of the disease we are studying. Now, for example, like gastric cancer is much more common in Asia. So naturally, you see more Asian patients in those cohorts. That is really an epidemiology issue, not a development limitation. At the same time, there is very much a global program. In fact, the head and neck cancer patient we discussed earlier who had this doable partial response on the HCB101 monotherapy, is actually from the United States. So I would say the current data have a strong Asian representation. But our goal is to build evidence across different regions and populations because ultimately, we want this to be relevant to patients globally. In fact, we will discuss our progress with the US FDA soon.
Okay, so thank you, Alvin, for the insightful deep dive. It is clear that HCP11 not only holds its ground scientifically, but is also demonstrating broad spectrum potential, advancing across multiple clinical fronts from gastric cancer and head and neck cancer. Thank you, Alvin. Anything you want to tell the audience?
So you asked me what can I add to the audience, Vivi. So toward the end, I just want one thing. One thing I really want the audience to know is that Taiwan absolutely has the talent and the scientific foundation to contribute to global biotech innovation. People sometimes ask, why Taiwan? For us, the answer starts with the team. R&D team, the research team in Taiwan has deep experience in biologic discovery and translational science. And that experience includes a real track record of successful innovation, like Shanghai Hines for Hanling PD-1, which was discovered by the same team here. Another strength is the depth of our preclinical platform. We have built a very broad tumor model system in-house, with more than 80 animal models, where most big pharma companies will have 3 to 5 animal models, which will allow us to test the programs across many different biological settings. That is important because drug discovery is not just about having a good idea. It is about testing that idea rigorously and understand where it is most likely to perform and to work. And of course, the real question is always whether preclinical work can translate into patients, into clinic. And that is why our early clinical data matters so much. I think the encouraging signal we are seeing at the beginning to show the science built in Taiwan can translate into a meaningful clinical potential. In fact, I used those animal data to support our recent orphan drug designation that granted to HCP101 for gastric cancer by the United States FDA. So to me, the bigger message to the audience is this. Taiwan is not just participating in the biotech innovation. Taiwan can help lead it, bring it to the finish line. Thank you for inviting me again.
Okay, thank you. 谢谢，感谢。